according to the 50 microliter reaction, since my buffer is 5x concentrated and I am setting up 50 microliter reaction, I have to use the 1x, right? That's the reason I am using of 5x buffer to make it 1x in the 50 microliter reaction, right? These calculations you have to learn before setting up anything. Please, this calculation, it is for everybody. Please just be confident that calculations you are doing, those are correct, otherwise you won't be able to set up the reaction, right? Make sure that the calculations you know by concentrations, not by microliters, please, right? That is a journal thing, mistake people do, that itna microliter dal diya tha. Please do not make a habit, always talk in the terms of concentrations, right? Because sometimes I may be giving you 5x or the 5x concentrated, and at other place you are getting 2x or 1x. So, you have to do the dilutions according to the concentration of your stock, right? And same thing is with DNTP, MgCl2 primer, because there is one stock concentration and what is your requirement, the working concentration, accordingly you do the dilutions, right? Dilutions you all know, N1, V1, N2, V2, right? So, you please make your individual dilutions in your notebooks just for the practice sake, and then you start doing it, right? कितना कितना डालना है इसमें?
नहीं, ये डाल दिया मैंने 120 के आसपास है।
हाँ, ठीक है।
5 किलो हो जाएगा।
ठीक है।
ये भी उसी के हिसाब से 60 डाल दिया। ये भी 15 डाल दिया।
हाँ, ठीक है, बहुत है। टैग अभी रखवा दूँ वहाँ पे, क्योंकि इन लोगों को तो अभी टाइम लगेगा ना, टैग
रख लेते हैं ना, इसी में ट्रांसफ़र करके रख लिए रहते हैं। नहीं, ट्रांसफ़र करके
टाइम लगेगा। अच्छा, हाँ, कर लो। 2 माइक्रोलीटर निकाल लो, या तो 2 बहुत होगा, है ना?
तो इसी में
या
2 माइक्रोलीटर बहुत होगा।
2 माइक्रोलीटर बहुत होगा।
अरे मार्कर इतना फेंट है, हाथ से मिल गया ना?
तुम्हारा हाथ कुछ पसीना वसीना आया होगा ना? पसीना वसीना आया होगा तुम्हें।
2 ना?
2 माइक्रोलीटर बहुत होगा ना, क्योंकि 10 रिएक्शन लग जाएंगे 2 से। कुछ रखा होगा उसमें, कुछ रखा होगा ना, मिनी कूलर्स। एक तो अभी कुलेश्वर वो मुकेश लेके गया, उससे पूछो मुकेश, एक मिनी कूलर जो तुम यूज़ कर रहे थे वो कहाँ? अपने और भी कोई यूज़ कर रहा है अभी हमारे लैब में? मिनी कूलर्स की ज़रूरत पड़, हाँ, एंगेज वाले ही बोल रहे हैं, बिकॉज दे रिक्वायर मिनी कूलर्स ना, देन हाउ दे विल सेट अप। Kavin, you are going there. Can you keep it in -20? Somebody is there.
Then you should tell him how did you come to this particular sample.
टैग कहाँ रख दिया?
टैग मैंने उसी में रखवा दिया, अभी मेरे कलर में अभी
ले जाते हैं ना, ये इसमें से ले
अच्छा नहीं, अभी कितने लोगों का हो गया? चलो मैं ले जाऊँ, मैं ले जाऊँ।
ये आप ले जाइए।
हाँ, ठीक है।
ये प्राइमर्स हो गए है ना?
ठीक है।
ये प्राइमर 1 2 3 प्राइमर्स हैं।
हाँ, गॉट इट।
बाकी ये डीएनए जिसका भी,
हाँ, ये मैंने डीएनए यूज़ किया है। ये फंगस वाला वो था ITS, ये मैंने पूल बनाया था फेंक से।
बस उसको रख दीजिए, बाकी
मैं ले जाती हूँ। ठीक, अब स्टैंड्स, PCR वाले स्टैंड्स चाहिए ना? सुनो, वो जो अभी वो आया है ना, अभी मंगाए हैं मैंने, नहीं अभी मैंने वो PCR के स्टैंड्स मंगवाए थे, छोटे वाले ना, वैसे मिनी कूलर्स नहीं है लेकिन वैसे स्टैंड्स हैं। हाँ, ठीक है। ये ना इधर आओ एक बार। एक छोटा सा मैंने डब्बा रखवाया था ऊपर, वहाँ पे जहाँ कुलेश्वर बैठता है ऊपर की तरफ, एक प्लास्टिक के जिसमें रैक होंगे, PCR रैक। एक ब्राउन कलर का ना छोटा सा डब्बा होगा, इतना बड़ा सा ना, जिसमें ऐसे वाले स्टैंड होंगे। ऐसे वाले
They have two samples. Sample number 1, the reading is 1.74. Right. Another sample, it is 1.64. The reading is so perfect. The reason is they used a Promega kit. Right? So let us accept it. And the yield wise, not bad, really bad. It's good. One sample, it is 128.8 nanograms per microliter. And another sample, it is 93.3 nanograms per microliter. So the problem is with the second sample, which is 93.3 nanograms per microliter. So they have no option now. They have to normalize the DNA template for 50 nanograms per microliter. Okay, since you cannot make 100 nanograms per microliter from 93.3 nanograms per microliter, so there is no other option now. You go for 50 nanograms per microliter, the template. Okay, do it now. You have to set a PCR. Okay. Any other group who has completed quantitation? Yeah, you forget it. That is for soil DNA. You don't have to bother. Forget it.
To give
Humic acid contamination. You forget it now. Okay. So not bad. The Magnaporthy DNA yield is invariably something like this only. Okay. Yeah. What about PCR for Magnaporthy? The protocol is done. Okay. Protocol is there. Let them start that immediately now without loss of time.
what madam told, madam ने जो बताया that you have written the calculation or not? Write down again in a sequence. You write down. I'll tell you. Like you write first in the extreme left milky water MqH2O you write.
What about other groups? What's going on now?
And then buffer MgCl2
So we have the results from two groups, group number 1 and 5,
MgCl2
right? So other groups still they are struggling with the system, I think.
primer forward, primer reverse, PF, PR, PF, PR, PR you write that is understood. Then your tag polymerase, then your DNA. So concentration already I told to all of you. So milky water is like this we used to write. Like we write it as C means stop concentration, as WC working concentration, then 50 microliter where our reaction. Suppose it is 25, we'll write here 25. Then what are the stock we are using? Like here madam told this is buffer.
We had these two readings, 302 and 184.6. So now we need 100 nanogram per ml DNA. So, and this is the volume.
Yeah.
So we'll do this only, na?
Yeah.
N1V1 is equal to N2V2. This is the N1 which is provided to us. How much volume we'll take of this? N2 is this one and volume is this one. This would be our working DNA. और इससे हम 1 ml लेंगे PCR करने के लिए, है ना?
You made it ready?
We're doing it.
Make it fast, don't drag it.
Reverse also the same. Then tag polymerase 5 units, 5 U capital U. Capital U. Okay. ठीक है. Then DNA. DNA depends. 50 we have. 50 you write. 100 है, 200 है, whatever you take. ठीक है. Then working concentration. You write down vertically 1x. Then 1x buffer also 1x. Then MgCl 200 micromole. Sorry.
If reading
This is
16.55,
1.5.
can we take 70 or we have to take 16?
Minimum.
17 you can take.
17.
Yeah, 17 plus
200
17 plus the rest should be buffer
Oh, yeah,
to
buffer.
make it 50. Yes.
33 microliter of buffer. Okay, make it fast.
Because 1 microliter is better. Picomole 10 to power minus 12.
Any group completed? Completed? Yeah. Show me the data. Group number?
10 micromole.
10 micromole it will be
This is Ralstonia? Pseudomonas. Okay. Now we just calculate for 100 nanograms concentration. You just calculate. Okay.
stock तो बनाते हैं 100 फिर उसको
Or you can go for 200 also because you have good reading.
Sir, the buffer which we are using it will be the same buffer
Same
which
buffer.
we extracted our
Exactly,
DNA.
yeah.
That is
Yeah,
true, sir.
last buffer. Yeah.
Sir, one thing I didn't understand that yesterday you told me about this thing. So this, that we use 10:1 and after that if DNA is less in amount then what's the logic behind
Yeah, we'll discuss the PCR. Na? We sum up today.
yes, sir.
picomole what happens suppose we are getting primer we have a particular volume of water to make it 100 picomole ठीक है per microliter concentration 100 picomole here 100 picomole हम बना लेते हैं then from here we dilute it to 10 ठीक है 10 times
So you do it for 200, right? Yeah, good.
from here 10 micromole
So we have a reading from group number 2. They have extracted DNA from Pseudomonas. I think they have a very good yield in terms of yield. It is 351 nanograms per microliter. Sample 2 is 220 nanograms per microliter. And very interesting 260, 280 reading, that is 2.12 and 2.14. So we need to discuss why this reading is beyond the standard value, what is expected out of this 1.6 to 1.8. We'll have some discussion. Maybe once everybody is through with the PCR, we'll do that discussion now.
10 you write 10 only. 10 only you write. 10 become 9 to 10 become. This is 0.2. Sorry, 0.2 will be 1 unit. 1 unit. Concentration is 1 unit. Concentration is ready for 1 unit.
So any more group is ready with the data? Mukesh? Now you can proceed with the PCR, right? Start now. So Bacillus group is going to amplify 16S ribosomal DNA. Yeah, start arranging the DNA and all the reagents. Yeah. Done? Lot of RNA. You didn't add. The gel will discuss that.
That's the same. Where is the scooping? Scooping. Where is the scooping? There it is 40, 137, 156, 101, and this ratio is this one. And the same after scooping, isopropanol, we went for centrifugation also. And there also we got this one.
Okay, fine. So, what is PS?
Pseudomonas.
Pseudomonas group, they had some other reading?
Same.
This is,
was the reading.
okay. This is scooping, okay, fine. All right.
Scooping one, maybe sir, it is not properly suspended, otherwise quantities would be more.
No, problems are different, okay. Yeah, this is group number 3, right?
2 and 3 both.
So, group number 2 and 3, we have the readings now. Group number 2, Ralstonia, right? Group number 3, Ralstonia, we have 40 nanograms per microliter and 137.2 nanograms per microliter. That was done with what is called as spooling out the DNA. With the centrifugation, the good reading 231 and 386. Pseudomonas group 351, 220 and 156 and 101. But by the by, how did you segregate 2 samples? You handle only 2 samples, correct? Now it becomes 4 samples now.
2 samples only, sir. 4 it later on it becomes when we 2 went for scooping and 2 went for centrifugation. The same 2 tubes which we were handling. So we scooped new tubes we have created, and the old ones we went for centrifugation also. The old one isopropanol step, at isopropanol the scooping we did, and 2 new tubes we created for 1 new tube for each sample we created.
Once you spooled out, what was the need for the previous tube?
Previous tube, that is what we thought, whether we are getting some difference here or not.
Yeah,
For that.
that is really creating a lot of confusion now.
No, sir, not confusion.
Yeah, it is a confusion. When you spooled it out, you should spool out every DNA.
Sir, whatever that pellet was visible,
That is what I am telling. It is not completely spooled out. You have taken a little bit here, and the majority you left it here, and in both the tubes you have DNA now. Okay, all right. So now the question is, you take one from here for your PCR. Let's see what is going to come out of it. Okay. Yeah. Now you calculate 137.2. You calculate for 100 and 386, the corresponding one. You take 100 here, and this is already maybe 100, and here also you make 100 and go for PCR. Okay. Pseudomonas, Ralstonia, Kuleshwar, for Ralstonia which primer? All 16S only. Only Magnaporthy is a housekeeping gene. You want to do it? Protocol? It works, huh? Great.
Chemistry is same, but reaction condition may be
No, she might have given the same. She told me also. Fine. So that means Ralstonia and Pseudomonas will go to the same PCR mission. Okay? But the Pseudomonas will take 16S ribosomal amplification primus, that is 27F and 1492R. Right? And for Ralstonia, it is a FLE-C. Yeah, please check it. One of the housekeeping genes of Ralstonia you take, and these two samples will go to a single PCR mission. So now one group is left out now. They are still struggling, it seems. I do not know what this group is really isolating. So that sample is, what is that sample? Sclerosium. Okay. No, don't worry. You go ahead of doing PCR and see. Okay, that is ITS. So we need 3 PCR machines. What is our plan, Kuleshwar, for 3 PCR machines? Okay. All the 3 machines are available, right? Yeah. It's available. Okay. Yeah. Now the 3 PCR machines are needed to do this 4.
now 2 only is available. One is already started.
Yeah. 2 more are there. So one is in lab number 10. You can set up maybe the 16S and the housekeeping gene of Ralstonia together, and ITS for Sclerosium can go to Dr. Deepa's lab. Okay.
Both the gene, Magnaporthy specific gene also has same
As ITS, right? So that means Magnaporthy and Sclerosium will go to the same machine, and Pseudomonas and Ralstonia will go to a machine that is a Pandap machine, and the Bacillus is already going on in this lab. Okay, that's great. So 5th group, what is the reading? You would like to know? Quantitation? 4th group. Again you extracted. Okay. Okay, fine. Okay, so you. Okay, what is the reading? Give it to me. That's what I was waiting here. So the 4th group has come out with the reading 462.4 nanograms per microliter and 721.8 nanograms per microliter. You make 200 nanograms per microliter stock DNA and go for PCR now. Okay.
PCR, whatever what PCR is running now.
Yeah, 260 to 80 ratio also. Not, it's very good, 1.79, and the another one it is 1.53. That is slightly on the lower side. Okay. Now you set up PCR. After PCR.
PCR is for this sample only.
Okay, this sample only. Okay, with ITS and 1 and 4. Okay. All right. Okay. So that means 2 PCR is going on here, right? Okay. Bacillus also.
No bacillus, only sample.
Okay, fine. All right, I got it now. So 4 samples need to go for PCR now in 2 machines.
Live? It's not a live coverage, I think. Live is going on. Just I checked, it's not coming.
Here I checked that is
Yeah.
if you have the link, if you are clicking that is going to the home one.
Okay. Because just now I checked, it's only recorded, it's coming.
ये लाइव स्ट्रीमिंग ना एक बार आईआर के ओपन करी आईआर वेबसाइट लाइव चल रहा है ना
I think live in YouTube, but IR it's coming as a recorded one.
the link is going to YouTube old record here it is coming live only is coming
सर ये कैलकुलेट कर लीजिए चार्ज इसको 4 से कैलकुलेट कर दें
Kumare? No, he has come here. Just make it come so. You can view there, it's live streaming. From here. Yeah. Check on the Govindaraj. It's only coming for the thing. Morning, recorded one. Siri, just I'm leaving now. Just I came to see the update.
रखेलो भी आएंगे सर तुमको लेना कोई सर आएंगे उनको लेना या हो सकता या तो वही होंगे नाम तो पता नहीं है
अपलोड करना पड़ेगा रिकॉर्डिंग हो रहा है ना
भैया इसको इधर कर लो यार ऐसे इसमें आ रहा है इतना हैजार्ड है और रिकॉर्डिंग मत करिए बंद कर दीजिए सही कर रहे हैं बंद कर दीजिए
चल रहा है
गंदगी से कर रहे थे क्या चला रहे हैं उधर हमने 15 रिएजेंट डाल दिए हैं दूसरा है हमारा बफर है दो सैंपल कभी मिक्स करने प्रॉपर्ली इसको मिक्स करके सेंट्रीफ्यूज आप पहले मिक्स करके हो गया क्वान्टिफाई? आ रहा है सही? कितना कितना? 450? ऐसा हो ही नहीं सकता, वो मशीन खराब है। सही बता और हाँ डायरेक्ट इसी से लगाएंगे, डाइल्यूटेड वाले से लगाएंगे। वो खराब है मशीन, सही बता रहा है। अब बताओ, जब ओरिजिनल वाला नहीं था तो डाइल्यूट करने के बाद कैसे बढ़ जाएगा? ये मशीन है। दोनों 16S है। 16S है उधर।
आपने डाल दिया सर?
हाँ कर दे।
नहीं सर, लास्ट वन ये
Here sir said 16S is okay. 10 minutes earlier sir said it's okay, 16S only. For both, for Rostonia.
For both
Not for primer also they said. So where is the primer? You can set it again. Cherry. Rostonia में कौन सा primer? प्राइमर क्या करेगा? दूसरा स्टॉक बनाना पड़ेगा सर। हाँ बन जाएगा, क्या करना है? इसमें ये कौन डाला है भाई, यूज करके? कैलकुलेट कर लो, दो ही रिएक्शन, दो ही सैंपल ना है? तो कर लो कैलकुलेट।
डबल सेम रिएक्शन।
इसका हाफ कर दो। पॉइंट एक का एक्चुअली कर लेते हैं, ठीक है? उस पे आ गया है
65.6 और सारा हो जाता है।
इसमें तो कुछ है
65.6।
एक काम करो, 16 भी सेट कर दो और वो भी सेट कर दो।
दोनों
बस सर को नहीं बताओ। बन गया तो क्या करें इंग्लिश मास्टर में इसका आप लोग 16 यूज कर रहे हैं 16 है इनका फिर यूज़ कर लेता हूँ इनका डीएनए ठीक है I will set this one as per you also because master वो मास्टर स्टॉक नेक्स्ट वाले हो ना listen वो मास्टर स्टॉक जिसको डाइल्यूट करके रखोगे ब्लड को डाइल्यूट किया होगा
सर डायरेक्ट एच नहीं करूँगा
हाँ मैंने इधर साइड में लिखा हुआ PCR ट्यूब्स लिखने का स्पेस नहीं ऐसे मिनी कूलर लाना पड़ेगा एक्चुअली I'm keeping it. क्या 10 माइक्रोलीटर इसको यूज करो फॉर यू पीपल 65 है
ये चल रहा है ना सर अभी चल
स्टार्ट है अभी ये टू मतलब क्या चाहिए UNTP एंड सेम नहीं है, इसलिए तो 2 बार बनाना पड़ रहा है ना। ये फॉरवर्ड है TTS कहाँ गई चाहिए, यही है ना? सर इसमें ये है ना राजकोनिया में
फॉरवर्ड 57
इसका 57 रखना है। मार्किंग कर दो
सर, ये मार्कर करो।
मार्किंग
राजस्टोन है सर, राजस्टोन वो भी है
हमारा है ना, राजस्टोनिया का नहीं है अभी। 2 का इसका करो, स्यूडोमोनास स्कूपिंग वाला जो साइन था और स्यूडोमोनास नॉर्मल था, ठीक है।
साइड में भी लिखो।
हाँ साइड में वो रेड वाला होता है। इसका मास्टर लेक्स कहाँ गया? अभी जो बनाया ये एक्चुअली उसमें होना चाहिए। चलो जल्दी से करते हैं। इस 2 को और लिखो तुम अपने लिए।
ये 2
ये राजस्टोनिया का है।
हाँ।
ये 2 इनका लिख दो। बैसिलस का है, बैसिलस का बस बैसिलस। So thank I'm making for you chicken good third, huh? Okay. हमारे ग्रुप का हमारे ग्रुप वाला ये स्कोप है, ये तो राजस्टोनी है, हमारे ग्रुप वाला भी है ना। हाँ यही तो था,
So we have different kind of experience, right? Diluted one, undiluted one, poorly handled samples, nicely handled samples, kit, laboratory, one stop, everything is there. Use this one directly. Choice of the biological material, how much content we may go wrong with. So 100 nanogram is the ideal. I'm sorry, 100 milligram.
these people कहाँ है कि notes दिया इसका bacillus
Making mistake is always good because that gives you a number of opportunities and you gain a lot of experience. Yeah, fungal DNA isolation. The secret is how nicely you are grinding.
First time working. Right now.
That is another problem.
Sample first time.
That's the kind of
Means one is
one reason. The another reason is the polymer.
मैंने 1 2 9 लिखा
That is difficult. That is difficult to reach. Complex carbohydrate is very difficult.
सर
That's the reason that is so far very young PCA, which is around 72 hours old.
इस पर 1 2
And enter into different developmental process.
1 है सर
So young mycelium,
इस पर 1 लिख दो
which is around 72 hours, is very ideal for this kind of fungus only. Activate is typically within 24 to maximum 24 hours. No, protocols are available. In fact, one of my projects we handle the DNA isolation from iridoscope. No mycelium at all. You have only iridoscope, and it's very difficult because iridoscope is having a thick coat with accumulation. You have to break what we
सब ऐड करने के बाद में इसको
then
टैप
we
करना
grind it for
प्रॉपर्ली
5 minutes continuously.
टैप में से
With the machine.
इसके बाद फिर इस पे
We have a handheld machine. You put the vessel in the front, just press it, goes around 5,000.
राजस्टोनिया के लिए 57 बोले हैं ना
हाँ सर
Put around
फिर कोई दिक्कत है
These are told to keep together.
हाँ 58 पर
सुडावाना सर इसको एक ही साथ रखें
200
58 पर रखना पड़ेगा फिर
mycelium.
इसका 58 है
You should
Then you just continuously grind it for about 5 minutes. So that gives you a complete homogenization of your mycelium.
Yeah, young mycelium is of course always preferred for DNA isolation, whether it is a oomyces or it is a fungi, different classes of fungi, maybe ascomyces, basidiomyces. The young mycelium up to 72 hours of age is best starter. Okay. Second thing, normally we go for the culturing in a broth with a constant shaking of 150 RPM for about 3 days. And you will see the fungal balls which is emerging, right? And take out the fungal balls and go for sterility check, as well as of course it is coming from monosporidial origin. Then the mycelium is harvested and dried at 60 degrees Celsius for 3-4 days in a kind of the Petri plate. Then it's a dry weight, right? Then that mycelium is taken and put it in the Eppendorf. Then you put glass powder and grind it with the machine, or optionally you can go for liquid nitrogen. You add liquid nitrogen, then it becomes so brittle and become powder, right. So, then you add the lysis buffer, then you incubate it, then you make a homogeneous lysate, then clean up step, and you go downstream. Okay. For spore, it is slightly difficult because the spores have to be ground thoroughly using a very strong abrasives. That is what we use, a sterile glass powder, right. Remember, glass powders are made in the laboratory, right. You take any new maybe a broken petri plate or some flask, you just grind it in pestle mortar. After 1 or 2 hours, you get a glass powder, right. It is not purchased, right. It is made in the lab, right. And we go for what do you call, you just put it in autoclave for a couple of days, 2 times you do sterilization, the sterile glass powder is always there. Okay. Keep the glass powder in -20 for many days, just to avoid any dust coming into, for that purpose only. Then whenever it is needed, you just take out, add a pinch, there is no measured quantity, you only add little pinch into the petri plate. With that machine, you get excellent results, there is no problems. Okay. That is what I am telling, the sample preparation is the most difficult, especially in the case of microorganisms, whether it is a fungi or it is a bacteria. The sample preparation is the key for successful DNA isolation, right. So, morning also I highlighted, you have to harmonize, you have to have a uniform quantity of the starting material across your samples. If you are handling 15 isolates, all 15 isolates, you make sure that it is only 100 milligram to 200 milligram of mycelium. If it is a bacteria, it is 1 ml, 1 OD, right. This you cannot deviate. Okay. Even if it is spore also, you must evolve a strategy to have a same quantity, same quantum of spores. For example, some fungi like Puccinia, you will never get a mycelium. The fungal isolation is always from spore only, because spore is the one which you can get it from the sample. We cannot culture this. So, you take the wheat plant and take it on the blotting paper, just tape it, the spores will fall. Then you gather the spore and you can even quantify. You can take the weight of the spore, right, 50 milligrams, 100 milligram. So, the quantitation of biological material is not that difficult, right. So, the uniform start is the key to very successful DNA isolation. Never ever take unknown quantity of biological material for DNA isolation. It is always a known quantity. Okay. So, do not ignore this, the sample preparation step. Many times we have the tendency, because you are always living with that fungus or bacteria, so you feel okay, I have the culture, I can take it. It is not like that, right. You will get some DNA, but it creates a lot of complexities and different readings you will get, and sometimes that upset your program. Suppose if you want 200 nanograms as a base, one of the sample is below 100, then it upset your entire programs, right. So, ensure that you have a good quantity DNA isolated, at least in excess of 200 nanograms per microliter, and devote around half of it for your experiments, and the remaining you keep it as a backup. And somebody was asking, a mini prep I want to use one time and I want to forget it. So, nothing like that, I won't agree to that point. So, if you are generating the data and ensure that the corresponding biological material at that particular point of time is preserved. That you may argue that I will get this virus once again in another time point, but you cannot argue that you will get the same virus once again, right. It is difficult always, right. That is what logically whatever data you are generating, the corresponding biological material need to be preserved, right, because the data is going to be preserved, right. And data preservation, we have the data centers. Yeah, you divide. So far people have not done, does not mean that you cannot do it. This is what I have been telling in the last 5 years. Viruses can be preserved, right. You extract virus, half of the virus you preserve it, what is the problem? Purify it, and purified virus you preserve it. So, unless you have the biological backup of your data remains still what do you call it is not really useful in the long run, right. So, that is what most of the laboratories whatever the sequence data they generate, from where they are generating the sequence data, the biological material is preserved. So, normally what we do, once the organism is isolated, you confirmed its identity tentatively and you found this is the one you wanted to isolate. Having isolated it, now the next step is you have to preserve it, right. When the preservation stage comes, now the question is how to preserve it. So, I do not want to comment on that. Fungi is prepared in a preserved in a different way, bacteria is preserved in a different way, maybe viruses may be in a different way, but whatever methodology you adopt, not a problem. But what is more important here is the material which is going into the preservation, right. You have to divide the material into two. One goes to preservation and the other half coming from the same biological origin should go for data generation. Remember, it is not another colony which is preserved and you take another colony for data generation, never, right. You spot the colony, this is the Pseudomonas fluorescens colony I am going to identify. If you decided that colony need to be preserved and a portion of that colony should be subjected for identification effort, whatever may be the effort, maybe a biolog or FAME or sequencing, a single gene or MLST or whole genome, whatever it is the same colony that matters, right. If you take another colony from the same plate, you think okay, I am taking from the same plate, both the colonies are looking alike, no problem, I will take one colony for preservation and another colony for identification, or sometimes you have the tendency of making multiple preservation. You pick around 3-4 colonies from the same plate for preservation, but chances are that all the 4 colonies may be different lineage, right. If fast evolving organisms, maybe it is a different lineage, or if it is from environmental sample, it is even more complicated. So, ensure that you handle only one colony. That is what from the original plate, the single colony preservation is still one of the best techniques. No other technique is beaten this technique now. The single colony is multiplied on the second plate. Ensure that the second plate is free from any other contaminants. And from that plate, you take one share for preservation, make another one for identification by biolog or FAME or 16S. That should be the strategy. Okay? Even if you are making multiple preservation, multiple tubes, normally we have in our laboratory, we need to go for 4 different tubes. All the 4 tubes should come from the same colony, right. So, all of them are genetically uniform, genetically identical. This is one of the most important criteria you have to adopt in your experiments.
This may be like this may hold for only bacteria.
No, even for fungi.
For viruses, I'm asking, like from a single leaf, we'll get quasi-species. Like it will be
preserve as a together. I think where is Charishma? She preserved the entire microbiome of Phyllosphere. Right? At least the origin from where you isolated is preserved. In those cases, you understand the entire diversity, what we call microbiome, or in your case it is virome, is preserved, and you expect that your target is still there. It is possible, right? Purification steps, if it is available, you purify and preserve it. If it is not available readily, it is always a mixed culture, you preserve the mixed culture, right? Otherwise, your data will be there, the corresponding biological material is not at all available. That is the it is not really scientifically correct one. Okay? In fungi, it is again everybody is taking effort to go for monosporidial lineage, monosporidial progenies, right? And we have beautiful techniques are evolved of late. It is coming the spore preservation in filter papers, and that is emerging as one of the very nice technique for long term preservation, right? So, anybody is working on fungi here, you take the conidia, for example, Magnaporthy we have developed in the laboratory for long term preservation. You grow fungus for about 5 to 6 days, and you just drop the filter papers filter paper, I mean Whatman papers. You just make a small disc out of it, and you just go for autoclaving, right? Maybe you can soak it in sterile water a little bit, and you autoclave it. And the autoclaved filter papers, the dry one is spread on the petri plate, right? And the fungus is allowed to grow for another 2 more days, right? And after 7th day, you start picking those filters. All the filters are picked using a sterile forceps and put it in the Eppendorf tube, right? And that Eppendorf tube is subjected for vacuum drying for 1 hour. Okay? So, and this you repeat 2nd day and 3rd day, right? On 4th day, you take the entire setup to flow cabinet and slowly release the vacuum, and without spoiling the sample inside, and take out the tube, and you will see the dry filters which is coated by the fungal spores, right? And this filter is preserved in -20, and any time you can retrieve that, and each disc is loaded with the spores, and you just spot it on the plate in the center, and within another 3-4 days you will see the beautiful colony emerging, right? So, this is the method of preservation. So, remember, the plate is coming from a single spore, and portion has gone for this preservation, another portion has gone for sequencing efforts, and the whole genome what we did for Magnapurthi, we have preserved like this. The same genetic material, half of the material has gone for genomic DNA isolation and packed bio library and sequencing, Illumina library and sequencing. Same biological material has gone for long term preservation in filter paper that was kept in -20 as a long term culture. The idea is you do not have to repeatedly subculture this fungus time and again. Remember, repeated subculturing makes the fungus different from the original, because there is a saying, every copy is less than its original, correct? That is true even for biological material, right? If you start putting the culture on plate, one plate to another plate, from 2nd to 3rd, 3rd to 4th, which makes the fungus adapted to the agars, and you may not be surprised at one point of time it may lose its virulence. It is well worked out in somebody handled Ralstonia in your group, it clearly worked out. On 4th and 5th subculturing, it is no longer a pathogenic Ralstonia. It loses its virulence once for all, right? Although some reports are there, it reverts back to the virulent form, but the consensus is it is irreversible phenomenon which happens with repeated subculturing. So, if you read the biolog identification scheme, they always say you preserve the original culture. That is very, very important. Original culture means the one which is isolated first time from the environment. Environment can be anything. It may be agricultural environment or whatever, soil or irrigation water, whatever it is. The first culture, whatever you get it, you may get some contaminants. From that plate, you make a fresh one. The second plate is invariably the pure culture. That pure culture should be preserved, and that pure culture should be subjected for sequencing efforts, because you have the entire function intact there, without much of alteration. Some alteration would have come there also, but not major change, right? So, if you have any quick wrap up here, it's all done PCR now. Okay, before that, if you have any quick, since you opened up this particular discussion, we entered into the discussion. If you have any quick questions, we will wrap up this today's session now. 260 by 280 ratio. What I experience, I can tell you. If it is, the reading is less than 1.6, you take it for granted it is having protein contamination. Okay, if it is more than 1.8, most of the time it is attributed to phenol contamination, which comes from the environmental sources, right? And we have a remedy also. If it is less than 1.6, you go for one more round of chloroform isoamyl alcohol treatment. Okay, try to remove as much of protein as possible and try to reach the value which is 1.6 to 1.8. The question comes, If it is above 1.8, sometime it is above 2 also, then how to address that? So, literatures are there, people have experienced it. The best method to remove the so-called invisible contamination which is attributed to phenols is diethyl ether treatment. Okay? So, you just add diethyl ether, equal volume of whatever you have in your tube, form emulsion. It's very difficult to form an emulsion with the diethyl ether because it's a extremely polar compound, so, and volatile compound. So, I have my own personal experience. You add diethyl ether, you form an emulsion, you centrifuge at room temperature. When you open it, you see only your aqueous. Your diethyl ether is completely evaporated, right? You do it at low temperature and try to remove that. You may get some reduction in your ratio. This is one remedy, right? Most of the time it works. Diethyl ether treatment will remove some of the contaminants which is increasing the ratio. You can try with one of your samples, somebody got reading 2 and above, you can try it with that. Okay. So, any other quick questions?
So, whatever we quantify today would not remain the constant when we do it again if we quantify the other day. So, what should be the probable reason?
Yeah, it's a very good question she is asking. What is quantitated today is not there tomorrow. Okay. So, the reason is your DNA was not dissolved properly. Okay. I probably have used water to dissolve so much of DNA, right? Small quantity of DNA dissolving in water is okay. If it is a big quantity in excess of 300, 400 nanograms, it takes time. Yesterday also I told, it takes efforts. For example, you need TE 10:1. Somebody was asking what is TE 10:1. 10 millimolar tris and 1 millimolar EDTA, 37 degrees Celsius incubation should be sufficient, and overnight at 4 degree, I am sure your DNA will be completely dissolved, right? And if you have low quantity, and there are some protocols which cannot tolerate your EDTA, we discourage this EDTA in those protocols. We go for EDTA minus buffer or less EDTA buffers. That is what I prescribe. TRIS 10:0.1, 10 times less of a EDTA. That is another option, right? Third option is you can go for only TRIS, right? And some people even prefer to go for water. Remember, the stock DNA is in TE 10:1 only, right? From there you make a working DNA in water and you spend it. So, that kind of DNA you do not make too much of quantity, you make around 10 microliters or 20 microliters and do your experiments, and do not try to store it for future backup, no. So, use it, throw it. You have always backup in 10:1, anytime it is needed you take it. Okay. So, we have 4 choices now: plain water for instant use, short term preservation in TRIS or TRIS 10:0.1, long term preservation it is TRIS 10:1. Okay. Dissolve DNA completely, otherwise the density gradient forms and you are taking 1 microliter out of 100 microliter of DNA stock, right. Definitely the chances of error is plenty here. If it is not dissolved, the heavy will be in the bottom and low will be in the top, and you take not a true representative of the volume which is really needed for the experiments, because you believe the machine's data, right. So, it should be a homogeneous completely dissolved DNA. Without that, I do not think you can perform any experiments. Okay. Anybody else? Anything else? Question? Any other quick? Okay. If no more questions, today's practical we are going to wind up by setting up the PCR. Okay. Once it is machine is on, you can leave today and come back tomorrow at 9:30, right. Then let us assemble in auditorium. We will have a quick wrap up of what we have done today, and at 10 o'clock we have a very nice lecture coming up, that is about single molecule real time sequencing technology, that is a pack biotechnology. Rakshit Chaudhary is an expert on this particular subject. He is going to give a talk. So, you can interact with him. Whatever doubts you have about single molecule long read sequencing, you please talk to him and get all your doubts clarified. That lecture will be followed by another lecture by CLC Genomics. And the CLC Genomics lecture is the direct sequel to what Anirban spoke this morning, the machine part or the software part. The speaker Jyoti will be speaking about that. These two lectures are scheduled tomorrow, and we will come here tomorrow afternoon here, and it will be the bioinformatics practicals, right. So, what I will do, the PCR product and your genomic DNA need to run the gel, that you do it tomorrow, and by 3 o'clock we start our bioinformatics practical. Bring your laptop with internet connectivity or hotspot, and some links will be sent to you tomorrow. Download all the links and the programs which is there in the link that is needed for you to carry out your bioinformatics practical tomorrow and the day after tomorrow. Okay, we are going to teach you how to read sequences, how to curate sequences, how to get the best sequences out of the trace file, and how to do the end trimming, assembly, notations, and how the sequence can be put it in the file for submission to GenBank or other databases for getting accession number. So, that practical will go for 2 days, tomorrow and day after tomorrow. Okay. So, with that we will conclude today's session, and I think it is still live. So, we will close today's session with this statement. Okay. Anything else? Deepa?
One more thing I just want to add to the biological material is very hot. If you want to publish a new species, something like that, and you have not preserved your biological material, after some time when anybody else they repeat the same work and they won't get your biological material intact, they will not accept the data. And whatever you have worked for 20-30 years, that will go waste because they are not getting back the biological material. So, in case of fungi and in case of any other organism, it's important, it's really very important, because type and X type we always want to preserve. So, whatever you are working, that has to be preserved first, and then start working on that. So, that thing I want to add.
That is a very nice statement. So, it is for biological material need to be preserved. That is what the culture collections have cropped up in many places, and people are giving so much of importance to culture collections.
Because I have an example like recent systematics new phylogeny came and new classification came on the basis of molecular phylogeny. They have not accepted many of that phylum as well, because Zygomycetina is not accepted now, because whatever the parameter, whatever the material they have worked, that was not preserved. So, since they have deleted this Zygomyceta itself, long many genus are not accepted, since that they don't have any backup, even though they search for X type also, but they could not get. So, even the type and X type, they will not get anything. So, that is very important now onwards. So, if you are working any aspect of fungi, bacteria, viruses, keep the biological material ready. Thank you so much.